Good evening, everyone. So today we're going to be talking on bacteriophage. I promised us yesterday that I want to treat this topic because most of the past questions that I actually looked at, they have not asked. Most of them asked questions on bacteriophage. So I know the normal routine these days for certain exams, they don't repeat questions. So we are just hoping that maybe question can come from here, and if it comes, don't want any of us to um lag behind. So we have something to write down, okay? So let me just quickly run through the bacteriophage, the cycle of bacteriophage and every of its components that we need to know, right? So first of all, what is a bacteriophage? They are bacterial viruses, okay? They are the viruses that infect bacteria, just in a simple definition. Bacteriophage are viruses that infect bacteria. So they are obligate intracellular parasites that multiply inside bacteria by making use of some or all of their host biosynthesis machinery. So they're also called phages. All right. So by being obligate intracellular parasites, it means that they solely depend on the host machinery for them to be able to replicate. You know how viruses, um, the major mechanism of growth or replication is that they majorly depends on their host um, genetic materials to be able to replicate. All right. So just like bacteria attack humans, there are also some viruses that attack bacteria, and that's what we call the bacteriophage. Okay. So they occur widely in nature in close association with bacteria, and they are distributed widely in the soil, feces, and in other substances in the environment. So they're associated with transmission of genetic material from one bacterium to another. Okay, so just a brief history for us to know how this bacteriophage came about. The activities of bacteriophage was first described by Twalt in 1915, who described it as an infectious agent that distorted the appearance of colonies of staphylococci. Okay, so it was first discovered in the colonies or when they were culturing staphylococcus species. So subsequently, the error in 1979 demonstrated the lytic activities of the culture filtrates on bacterial colonies. So he suggested that um this agent that exhibited this lytic activity must be a virus. That's why he gave the name bacteriophage. So phage means to eat. Then bacteriophage, it means something that eats bacteria. So they are viruses that eat bacteria, just in a layman's understanding, all right? So we have the morphology, like the structure of how this bacteriophage looks like. So most of these phages, like I say, you can call it bacteriophage or you can call it phages, okay? Phage. They consist of single linear and double-stranded DNA genome, and the genome is surrounded by a protein fold known as phage capsid. If you are here when we treated the different viruses, we notice that one of the core components of a virus is capsid, viral capsid, that helps to protect the genome, maybe RNA or DNA. Okay, so this phage also contains what we call a phage capsid. So large phages usually consist of a head and a tail. The head encloses the genome, that's what the genetic material is. And then the tail is used as an organ of attachment as well as the conduit, that's like a passage through which the phage DNA passes into the host cell. So these are just telling us how this bacteriophage looks like. And you can see from the picture, this is what a bacteriophage looks like. This is the head. And this red coil thing is known as the genetic material, DNA. And it has the hexagonal head. And then this is the consist of the passage that leads to the tail, okay? That helps in attachment to a host as the bacteria in this case. All right. So that's it about the morphology. Then let's talk about the life cycle of a bacteriophage. How do they be able to like invade the bacterial cell and then cause maybe lysis or any other thing that they cause to the bacteria? So phages exhibit two different types of life cycle. We have the lytic cycle, then we have the lysogenic cycle. The lytic cycle, also called the virulent cycle. Okay, so They involve intracellular multiplication of the phage, which results in the lysis of the host bacteria, okay, resulting in a release of progeny virions. So I'm still going to explain the lytic cycle more, but what is talking about the virulent cycle that involves like killing of the bacteria by lysing the cell wall of the bacteria. But in lysogenic cycle, which is also known as temperate cycle, the phage DNA becomes integrated in the bacterial genome and replicates with the bacteria synchronously without causing any harm to the host. Just unlike the lytic cycle that causes harm to the bacterial cell, the lysogenic, it doesn't cause harm, rather it will integrate its genetic material, maybe DNA, into the bacteria own, and then as the bacteria tend to multiply or replicate, it also replicates with it. All right, so that's the major difference between lytic and then the lysogenic cycle. Then let's talk about the lytic cycle. How does it happen? So lytic cycle involves like 5 stages. We have the adsorption of the phage of the bacteriophage. We have the penetration of the bacteriophage, synthesis of the phage components, maturation, and then the release of the progeny phases. So these are all the phases, the different stages that these viruses called bacteriophage use in um, attacking or invading the host, in this case, bacteria. Do you understand? Number one, they absorb, that's attachment. Number two, they penetrate. Number three, they synthesize the phage components. Number three, when these phage components are synthesized, they mature. And then number four, they release the progeny phages that causes the cell lysis. Okay, so let's talk about absorption. Absorption is the first stage and is a specific process. So it depends on the susceptibility of the bacterium to the specific phages. So the specificity of the phage is determined by the presence of chemical receptors on surface of the bacteria. So absorption there occurs in a random collision and is dependent upon the concentration of both the susceptible bacteria and then the phage. So it's a very rapid process. Another normal condition is completed within minutes. This is just trying to explain how these bacteriophages tend to attach or how it tends to um, get to fix itself on this bacterial cell. Maybe when this bacteriophage want to infect a bacterial cell, they are very specific and they attach themselves to a specific receptor. So if this bacteria have the necessary chemical receptor that this bacteriophage attaches to, it there will be adsorption, that is attachment. All right. So that's all this first stage is talking about. The next one is penetration. It involves the penetration of the phage nucleic acid, that's the genetic material, into the bacterial cell. Okay, so penetration to the cell wall is facilitated by an enzyme called lysozyme, which is present in the tail core that produces a hole on the bacterial cell. When you look at the diagram, I showed you the head and the tail part of the bacteriophage. So this tail bacteriophage, it helps in absorption, that's attachment, and it's also first hasten the transfer of the bacteriophage genetic material to the host cell. Okay, so the phage DNA passes through the central core into the host cytoplasm, while the empty protein capsid remains attached to the surface, the cell wall surface. All right. So this penetration just trying to explain how because the major aim of this bacteriophage, what they actually want to do, the major thing they want to do to the bacteria, that they want to transfer their genetic material into the bacterial cell, like into the bacterial cell, right? So if they transfer their own nucleic acid into the bacterial cell, the bacterial cell will then absorb it, okay, and it will integrate itself in the bacterial genome, right? So when it integrates it will use the bacteria genetic materials or components to be able to replicate its own. Like we said, they are obligate parasites. So they depends on the bacterial's genetic material to be able to replicate. So that's the major thing to do. Then when they were able to penetrate, the next step is synthesis of phage components. They will then start replicating, like growing inside the bacterial cell. So this is immediately after the penetration. Then we have maturation, when they are able to produce this the phage component, they will then mature. That's assembly of the phage component into the mature effective phage particles. Okay. Then after the maturation, we have the release of the progeny phase that we formed, all right? So new mature progeny phase are released by lysis of the bacterial cell, because when there's attachment, notice here when I was talking of penetration, it say the lys it produces an enzyme called lysozyme present in the tail core that produces a hole on the bacterial cell wall. So when they were producing a hole on the bacterial cell, when one produced two, three, four, five, six, like thousands of these bacteriophage produce for the bacterial cell, it will causes the bacterial cell to explode or burst. Okay, that's what we call lysis. All right. So and when um they call this cell lysis. That means the bacterial cell is being weakened and they will die off through lysine. So that's the stages of the lytic cycle of the bacteriophage. How they attach, they penetrate, they synthesize their own phage components. When they mature, they release this progeny phage that causes the lysis of the bacteria. I hope you are following. So this is just a diagrammatic explanation of what I explained. Number one, this is a picture of the bacteriophage. Then this is a picture of the bacterial cell, the one that looks like an egg. Okay, so that's the host, the bacterial cell. As we say that bacteriophage attacks or they eat, there are viruses that attacks the bacteria, right? So this attachment, so when the bacteriophage, you can see the head and the tail is using the tail for attachment. So when it attaches the bacterial cell, the next one is to penetrate by injecting, you can see the green or the bluish substance, that's the DNA or the genetic material of the bacteriophage. So it injects it inside the bacterial cell, all right? So after injecting it, they will then start reproducing. They'll start replicating inside the bacteria cell. You can see the multiple replication going on at the third stage, that is the biosynthesis. The phage DNA replicates and then the phage proteins are made. Then it leads to the fourth one, which is maturation. That is alignment of the phage, the proteins and the phage nucleic material. So that's the maturation. They assemble, and when they assemble, they then cause the bursting of the bacterial cell, which is called lysis. So the cell lysis releasing the newly made phages, and these newly made phages will go on to attack other bacteria that they are susceptible to. I hope you understand, right? So let's go back to um the second cycle, which is the lysogenic cycle. Lysogenic cycle, they involves infection with every phage does not, okay, infection with every phage does not result in lysis of the host cells. So unlike the virulent phages, which is the lytic phages we explained, which causes lysis of the host cell, the temperate phage or the lysogenic phase integrates into the genome of the bacterial chromosome without causing any lysis of the bacteria. Do you understand? So the major aim is to just integrate themselves into the bacterial chromosome. Okay, so the integrated nucleic acid is known as the prophage. That is the one that attaches itself into the bacterial chromosome. They are known as prophage. So this prophage, they behave like a segment of the host chromosome and multiply synchronously with it. So this phenomenon is known as lysogeny. The bacterium that carries a prophage within its genome is called a lysogenic bacteria. So what he's trying to say is that just like how like checking for illustration I'm going to use for this. So this in lysogenic cycle, so what is the bacteriophage under this that uses this cycle? What they do is that when they attach to a bacterial cell, they will come and they kind of adapt themselves or attach themselves to their chromosome. They'll make themselves to be part of the chromosome, okay? So that when the bacteria is replicating or when the bacteria is um growing and replicating, just like what I said, then they too they will be replicating alongside with the bacteria. They will not cause any harm to the bacteria. It's just that they'll be replicating with it, growing with it, the bacteria. They also remain dormant, okay? So that's what the lysogeny is talking about. Then this a pictorial representation of what I'm talking about. Like in the lysogeny phase, just like the lytic cycle, the bacteriophage attaches to the cell wall of the bacteria, so they infect the cell wall. And then the next step they did that they integrate themselves in the bacterial gene chromosome. All right. You see the integration, right? You can see the gluten attaches itself to the bacterial chromosome instead of them um to start replicating on their own, that is using the genetic material but to start replicating on their own and producing the variant phages. They just attach themselves to the bacterial chromosome. And then the next thing is that they start dividing. So when the bacteria is dividing, they also follow it and divide. Then under stressful conditions, these divided chromosomes or these divided the lysogenic cycle can be excised from the bacterial chromosome and enter into the lytic cycle. That is, they can be a kind of changed or they can be forced to enter into the lytic cycle. I'm going to explain this more in the next stage. So when they enter the lytic cycle under a stressful condition, then the phage, they'll start replicating and then the phage proteins are made. They'll just continue with the lytic um, cycle. Then after they start replicating their own genetic material, they then assemble, and when they say the causes cell lysis, releasing newly made phages. So under normal circumstances, the lysogenic cycle doesn't cause any harm, but under a stressful conditions, those ones that will be replicating with the bacteria, that's these ones, they will just shift themselves from the bacterial chromosome and then they'll start replicating on their own like that. The lytic cycle that we've explained, right? So that's how the lysogenic process takes. So what are the applications or uses of bacteriophage? Number one is that they convenient model for study of virus-host interactions. Since we say that they majorly attack the bacteria, so they are very good for studying how these viruses and then the bacteria interact. So they play an important role in transmission of genetic information by a process of transduction. So we are going to talk more on transduction as a way of bacterial genetic transfer, gene transfer. Okay, so they play a very critical role there. So they also use as cloning vectors for genetic manipulations. Yeah, when you want to maybe do some researches or carry out any study, so you can use this bacteriophage that affects bacteria to make that study, maybe any genetic manipulation you want to do, you can just use it. So they also use for phage typing, that is used in diagnostic laboratory for the identification of pathogenic bacteria. Okay, battery is low. Okay, so they can also be used in diagnostic lab for identification of pathogenic bacteria. And then this phage can also be used in treatment of bacterial infections. So there are a lot of researches that is going on currently to know if these viruses that actually kills bacteria, they can just be used. You know, antibiotics also kills bacteria. So if bacteriophage actually kills bacteria, so why can't they be used also to treat most of the bacterial infections that humans use? Because these bacteriophage, they don't the research that we made so far, they don't really affect humans. They are not susceptible to humans because most of the receptors that they are attached to is majorly found on the bacteria. That's why they majorly attack bacteria. So if they don't affect humans, all these bacterial infections that we suffer, why can't we use bacteriophage in treating them? Since a lot of bacteria are now being resistant. So the research is still going on, and then a time will come when the bacteriophage will be manipulated in such a way that it will be using just like antibiotics. Do you understand? So we have gene transfer mechanisms in bacteria. So this gene transfer, it allows bacteria to acquire new genetic information from other organisms, including those from maybe that's from not from their own class or family tree, leading to a different genomic structure. Okay, so this process enables bacteria to overcome environmental challenges and thrive in a diverse environment. So the major aim of the gene transfer is just allow different bacteria to exchange genetic material so they can be modified, all right? They can be modified, and there are different ways that this gene transfer can be done in bacteria. So we have three major ways or three major mechanisms. Number one, we have the transformation. Number two, we have the transduction mechanism. And then number three, we have the conjugation mechanism. So for transformation is the process of the transfer of DNA itself from one bacterium to another. And this can occur in a nature or in a laboratory. So when it occurs in a nature, the DNA is released from a bacterium, maybe by lysis, all right, which may be taken over a recipient bacterium that must be competent. And this natural process of transfer, natural process of transfer of genetic material appears to play no role in disease. So, so this is talking about maybe when a DNA is released by a particular material, maybe the cell wall of that particular is lysed. So the DNA is released. You know, when the cell is lysed, the cell wall is burst, okay, and they release a lot of genetic material. So maybe another bacteria will then naturally take up the genetic material. So this can lead to transformation we are talking about. So, but when it occurs in a laboratory, it means DNA may be extracted. So this is done, you know, laboratories, maybe you want to carry out a research and then you extract a DNA from a particular bacteria and introduce it into the genetic makeup of another material. Just introduce one the DNA of a particular material, just let's say E. coli, Escherichia coli, introduce it in a staphylococcus, all right? So the cell walls of the bacteria in vitro studies mean outside the body of the organism are made up of more permeable for DNA uptake by using substances such as calcium chloride. So example of this kind is Bacillus or Haemophilus or Neisseria, and then pneumococcus can take up DNA from the environment. And then the DNA that's taken up can be incorporated into the recipient's chromosome. So this transformation method is just saying that DNA can be transferred from one bacterium to another. It can occur in a nature, maybe naturally, or also in a lab. If we want to do experiments or a research, all right? We want to check if a particular DNA of a bacteria is taken to another bacterial genome or integrated into another bacterial genome. What will happen, or what's the result we're going to get, all right? So we have another mechanism, which is the transduction. The transduction is the transfer of a portion of DNA from one bacterium to another and is being mediated by a bacteriophage. So this occurs through the process of the lysogenic conversion, which confers a new property to the bacterial cell. For example, by lysogenic conversion, non-pathogenic bacteria can become pathogenic, just like bacteriophage encode cholera toxin, which can be transferred from one bacterium to another by transduction. So what this transduction is talking about, that this transfer of a DNA material from one bacterium to another can also be done by those bacteriophage, which are viruses that attack the bacteria. So they do this process during the process of that lysogenic conversion. So, you know, in lysogenic conversion, the bacteriophage integrates its genetic material into the bacterial chromosome. So by so doing, they tend to replicate, and then they can, that time they'll be separated from the chromosome, they can carry out some part of the genetic material. And then when they infect another bacteria, they can introduce it. And one of the major, maybe disadvantage of this is that a non-pathogenic bacteria can be pathogenic by this process. Do you understand? So transduction, it has two types. We have the generalized transduction, and then we have the specialized transduction. The generalized transduction is just talking about that any part of the bacteriophage, the gene can be transferred to another bacteria. Like any part of the DNA material can be transferred to another bacteria. Okay. So why the specialized transduction is talking about this is specific part of the gene that will be transferred to a bacteria. This may maybe because this is a specialized, or they are very susceptible. The bacteriophage and the bacteria are very susceptible to each other. All right, so this is about the transduction. So let's look at the conjugation, which is the last but not the last mechanism of gene transfer in bacteria. So what is conjugation? Is a process of transfer of DNA from the donor bacterium to the recipient bacterium to the mating of two bacterial cells. So there must be, in conjugation, there must be a donor bacteria and there must be a recipient bacteria. So the donor bacteria is majorly regarded as a male, while the recipient bacteria is majorly regarded as a female. So when there's conjugation, it's kind of them having this sexual transfer. Let me just call it sexual transfer of the genetic or DNA material. Okay, so in conjugation, there's direct contact between the donor and the recipient. Remember when I explained about transformation, this the DNA material can just be found in the environment, or it can be laboratory, right? In the environment, in the side that there can be a cell lysis, then another bacteria that is competent will then pick it up. But here, there must be direct contact between the donor and the recipient. Okay, so it's always the donor that transfers the genetic material to the recipient. So conjugation takes place between two closely related species and occurs mostly in gram-negative bacteria. It also rarely occurs in gram-positive too, okay? And then in bacteria, there are two mating types, a donor and a recipient, which I explained. Because the recipient lack a particular um, a particular receptor, which is called the F factor, that's why they are not able to donate, or they are not able to transfer the DNA material. But the ability of a bacterium to be a donor is because of the presence of the cell of an extra piece of DNA called the F factor, or the fertility factor, or the sex factor. That's why they were able to donate, because they have a type of a receptor or a factor that is called the F factor, or a fertility factor, or a sex factor. So the F factor is a critical piece of the DNA. So it's part of the DNA that can replicate autonomously in the cell. So it is an independent replicon. So extra chromosomal piece of DNA that can replicate independently are given the general name plasmids. So I don't know, do you understand at this point? Let me know if you understand, because I feel I'm speaking so fast. Just let me know if you understand at this point in time. Hello, can you hear me? Hello, Ijoma, can you talk to us? Jeremiah Darby, can you hear me at this point?
Yes, we can.
We can hear you.
We can hear
I
you.
can hear you.
Okay,
This is
so we
I
are
actually
can
my
hear you. I'm
first
just
time.
joining now, but I can hear you.
okay, so we are all following, right?
Yes.
We are.
Okay, that's beautiful. I just wanted to confirm. Okay, so that is it about the conjugation. So in conjugation, just note that there must be a donor and there must be a recipient, okay? And the process, there must be a contact between the donor and the recipient, okay? So, and the donor is able to transfer this DNA material because they have an extra piece of DNA called the F factor, or the fertility factor, or the sex factor, okay? And this extra piece of DNA, they are called plasmids. So let's look at what is plasmids. So plasmids are extra chromosomal genetic material, okay, that's capable of replicating autonomously or independently. So they can replicate on their own without needing the main chromosome, okay? So they are usually much smaller than the bacterial chromosome. So just note that these plasmids are found in the bacterial chromosome, all right? So they will not be mixed up. They are not found on the bacteriophage. They are found on the bacterial chromosome. So they are usually much smaller than the bacterial chromosome, varying from less than 5 to more than several hundred kilobase per kpp. So the plasmids can also be present as integrated with bacterial chromosome. Plasmids integrated with host chromosomes are known as episomes. They are present in both gram-positive and gram-negative bacteria. So we have different types of plasmid, and they can be classified based on the transmissibility or the nature of the factor. So these are the two major ways that these plasmids are being differentiated. Okay, so based on the transmissibility of plasmid, we have two types. We have the transmissible or the conjugative plasmid, and then we have the non-transmissible or the non-conjugative plasmid. What are the non-transmissible or the conjugative plasmids? They can be transferred from one cell to another, just like the name, something that is transmissible, something that is transferable. They can be transferred from one cell to another by a process of genetic transfer known as conjugation. You understand? Can you now relate when we're explaining the conjugation mechanism of gene transfer? So they are large plasmids and they have all the necessary genes necessary for autonomous replication and for transfer of DNA to a recipient. Example of this is genes for sex pilus, which facilitate direct contact between the donor and the recipient's bacteria, enabling the genetic enabling the transfer of genetic material during conjugation. So what is talking about is that in transmissive or conjugative plasmid, they are the ones that can be transferred from one bacteria to another, just like the name. So they do this through conjugation, and we explained that conjugation involves the direct contact between a recipient and then the donor, right? So the donor is the one that's able to transfer the DNA material to the recipient. So for the non-transmissible or the non-conjugative plasmid, they are the ones that cannot mediate conjugation, and they are usually smaller than the conjugative plasmids, and they lack one or more of the genes needed for transfer of DNA. So a non-conjugative plasmid can be transferred on conjugation if they also harbor a conjugative plasmid. This is just opposite of what I've explained. The non-transmissible is one that cannot be transferred from one bacteria to another, while the transmissible is one that can be transferred from one bacteria to another. I hope you understand what I'm talking about. And they can be able to do this, that the transmissible, because they possess a particular receptor, which is we call the F factor or the sex factor or the fertility factor. That's why the donor or the conjugative ones is able to do that. Okay, so let's talk on the classification of plasmid based on the nature of factor. Okay, so the plasmid can contain what we call the F factor. They can contain what's called the R factor, and they contain what we call the cofactor or the polysynogenic factor. All right, so these are the three major factors that a plasmid can contain. And when a plasmid contains F factor, it means that it's a transfer of a factor that contains the genetic information essential for controlling mating process for the bacteria during conjugation. Remember what I explained about the conjugation process by containing F factor. So they're able to do that because they contain this F factor that is essential for controlling the mating process. That's a sex process or the sexual genetic transfer process between the donor and the recipient. So, and when a plasmid contains what we call the R factor, which is the resistance factor, okay, the resistance factor, they are circular with double-stranded DNA, and they consist of two components, the resistance transfer factor, RTF, and then the resistance determinant, which is denoted with small letter R. So the RTF is responsible for conjugational transfer, while the R determinant, the small R determinant, the resistance determinant carries resistance for one of the several antibiotics. So when you hear that a particular bacteria is resistant to an antibiotic or a drug that is given to it, it can mean that that particular bacteria may contain what we call the R factor. It carries resistance for one of the several antibiotics. So the R factor are responsible for spread of multi-drug resistance among bacteria, and they carry the genes for a variety of enzymes that can destroy antibiotics or modify the membrane transport system. So the R factor may carry one antibiotic resistant gene, or they may carry two or more of these genes. So what are the modes of these resistant genes? What are the modes that this R factor is able to be resistant to these antibiotics? Number one is modification or detoxification of antibodies. Example is the beta-lactamates. Okay, they can modify or it kind of produce toxins that neutralize these antibiotics. Okay, and another way is alteration of the target site. That is the target that this antibiotic is meant to focus on. They can just alter the target site, then rendering the antibiotic useless. Alteration of uptake, that's the amount that this antibiotic can be taken. This can be seen in tetracycline resistance, and also replacement of sensitive pathway. Most of this pathway that this antibiotic, that is the route that this antibiotic is meant to take, this R factor plasmid can just replace it, okay? And then the antibiotic will not be able to follow the major pathway it's supposed to be able to be fertile or to be able to produce the efficacy that it's supposed to. Okay, so let's talk on the Hall factor or the hallucinogenic factor. So it's a plasmid that resembles the I factor in promoting conjugation. And sorry, I have to take this up. Distracting me. So the cofactor is a plasmid that resembles the F factor in promoting conjugation, leading to self-transfer and also at times transfer of segments of chromosomes. They cause for the production of what we call the colicins. Okay, the colicins are antibiotic-like substance which are specifically and selectively lethal to other enteric bacteria. So the cholicinogenic factor, the major aim is to produce toxins, like I say, it resembles that of the F factor in promoting conjugation, okay, leading to self transfer and also transfer of segments of chromosomes. So what they do that they codes or they help in production of different toxins that that makes bacteria, sorry, that make antibiotic that renders antibiotic sorry, they cause for the production of different toxins, which are like antibiotic-like substance that specifically or selectively lethal to other enteric bacteria. So these carcinogenic, they produce or they help in production of different toxins which can be lethal or which can kill other bacteria. All right. So that is it about the different types of the plasmid. So this plasmid that is being found in bacteria, what are the functions or what do they actually help these bacteria to do? Like we explained, they are incur resistance to one or several antibiotics because they are extra chromosomal DNA which can replicate independently or autonomously. They carry genes that can confer resistance to a lot of antibiotics. So they can just mention the different ways they can do this, maybe alteration of the site, production of toxins that can neutralize all this. So the function of plasmid can be they can incur resistance, making the bacteria not to be killed by a lot of antibiotics, a specific antibiotic, all right? So we also have production of toxins. This certain plasmid allow bacteria to degrade environmental toxins, which can be beneficial in bioremediation efforts, all right? This is just the advantage when it comes to environmental health. So you also have synthesis of cell surface structures. So required for adherence or colonization. So some plasmids contain genes that contribute to the pathogenicity of bacteria, increasing the ability to cause disease. Okay, like synthesis of different cell surface structure. All right, so let's look at okay, this is for self-assessment. All right, so for we've discussed about the bacteriophage. I try to make it as very short and straight to the point as much as possible because there's no time again to come and start cramming a whole lot of things. So by this time, I hope everybody know what is a bacteriophage at this point in time, and the different cycle of the bacteriophage, the lytic and then the lysogenic. All right. Do we all know? So this is just like a self-assessment that you can just answer for you to know if you have really understood what is being said. Then the temperate phage or the temperate phage. What's another name for the temperate phage? Can someone remember? Another name for the temperate phage. Can we remember another name for the temperate phage? Hello, can everybody hear me? Abi, we've forgotten. Hehe.
We have forgotten.
Wahala. You don't have to forget. Okay, so, and also believe that
Is the lysogenic? I
yes,
think
that's a
lysogenic or
lysogenic phage or temperate phage. Yes, you're right. Then we have the different gene transfer, like the transduction, the conjugation. Just know how to maybe what each one entails. Then what are the events that leads to lysogeny? I try to explain like viral attachment and entry integration, and then replication and progeny projection. All this can lead to lysogeny. All right. Then event that can lead to the lysogenic termination is in an uncomfortable or in adverse conditions, just like what I explained, that when this lysogeny, that is integrated bacteriophage in the bacterial chromosome, it can just be there dormant, but when there is an unfavorable condition like desiccation, exposure to maybe UV rays or chemicals, they can also lead to lytic cycle. We saw it in the picture or in the diagram that I showed us. So I saw this question: what is an operon? Operon is a functional unit of a genetic material in prokaryotes, like in bacteria and archaea, that consisting of a cluster of genes that transcribe together into a single RNA and then molecule encoding multiple proteins. So the genes are functionally related and regulated as a group, often involved in some biochemical pathway. So operons allow for coordinated genes expression, enabling bacteria to quickly adapt to changing environments. So what the major thing about this operon is, it's just a part of a genetic material in bacteria. Then it allows the bacteria to quickly like replicate. It's just that part that maybe that helps to make the bacteria to replicate fast or to grow fast in the environment, to be able to adapt to any environmental condition that it finds itself. So that's what a operon called. It contains clusters of genes that can be transcribed together into a single mRNA molecule, including multiple proteins. So it can encode multiple proteins to allow the bacteria to grow fast, to be able to replicate, and then quickly adapt to a specific environment that it found itself, right? So that's it about the operon. I think I saw it in the TMA of this particular module. So that's why I decided to put it here, just in case. So at this point, that's the end of our discussion class. And if you have any question, just ask. Okay? If you have any question at all.
Please, I have a request. Can you please show me the slide for lytic where you discuss about the lytic? Because my network logged me out. Please.
Okay, my lytic here. Is it the diagram or the write-up?
The write-up, it's logged me out. So I just reappeared when you were explaining lysogenic.
This is
Okay,
it.
yes. Thank you.
Okay.
Thank you. I'm okay. Yeah.
Okay, no problem. So if you have any other question, you can still ask question on the group. If I'm still awake, I'm going to answer it. Maybe if you're watching other content and other modules, then you come up or you're confused at any point, you can still ask in the group. Okay?
Please, I have a request.
Yes, ma'am.
Is it possible that you just, I don't know if it's possible, can you just briefly go through the malaria life cycle again?
Malaria life cycle.
Yes.
Okay. Where can we see that? Let me check. That should be parasitology.
Yes.
Okay. So, okay, I can just use. So the life cycle of malaria is it just involve like 5 major stages. Number one, must be presence of an infected mosquito. So if like ordinary mosquito that is not infected with these parasites bites someone, it will not affect the person. So there must be presence of infected female Anopheles mosquito okay, before there could be transfer or transmission of this parasite. So the first stage is mosquito bites and then sporozoite infection. So a female Anopheles mosquito is infected with this Plasmodium parasite. Then when it bites a human, it injects what we call a sporozoite, okay, into the bloodstream of a human. So when inject this sporozoite, this sporozoite is just a form of this Plasmodium parasite, okay? There is infectious stage of this Plasmodium parasite. So when it injects this sporozoite, when this sporozoite enters the body, it goes straight to the liver. That's what we call the liver stage or the exoerythrocytic cycle. Exo means that it's happening outside the red blood cell. Okay. Um, so the liver stage, the sporozoa travel to the liver and invade the part of the liver called hepatocytes, which are the liver cells. So this takes between 5 to 21 days. So inside the liver, these sporozoids rapidly invade the liver cells where they mature into liver stage chisons, which burst to release like 2,000 to 40,000 merozoids. All right. So this is just talking of what is happening inside the liver from the infection site or from the biting site. The sporozoid traveled immediately to the liver. And then when it travels to the liver, it infects the liver cells, making it to, when it infects the liver cells, the sporozoids will mature, then release what we call the liver stage chisons or the merozoids. All right. So when it releases this merozoid will then be transferred or enter into the bloodstream, which is called lead us to the third stage, which is called the blood stage or the erythrocytic stage. So this erythrocytic stage is when the Plasmodium has entered or has invaded the blood cells. So the merozoids are released into the blood and infect the red blood cells. This is when you start feeling the symptoms of malaria, like the fever, the chills, the cold, like any symptoms of malaria. At this stage, that is when the malaria has really affected your blood, right? So in the blood stage, there are two things that happen when the malaria affects the blood. They can either, or some part of the merozoites that are in the blood can either undergo what we call the gametocyte formation. So some merozoites in red blood cells develop into a sexual form, the male and female gametocytes. So I'm going to tell you what these gametocytes do, and then some they, these gametocytes that they produce, okay, some of them will then form what we call the the sporozoites. When the merozoites that are in the red blood cell, they develop into the cellular forms, and then inside the cellular forms, they will not be a kind of an effective stage. Okay, so that when other mosquito come and invite the person, it will get this gametocyte and it will start developing inside the bacteria. Sorry, it will start developing inside that mosquito again. I don't know if you understand what I'm saying, but let me show you another thing. So this is just like a pictorial of a human diagram when a mosquito, this is the mosquito trying to bite the upper arm of the person. So when it bites this person, it will transfer, you see the different sporozoid that is biting into the person. So this sporozoid will go straight to the liver. You can see the liver here. So in the liver, it matures and forms what we call a merozoid. These merozoids, we say it can be from like up to 40,000 merozoids, which are being released and then they enter into the blood. So inside the blood, some of the merozoids will start infecting the person. That's when I say that you start seeing the symptoms of feeling the symptoms of malaria, like fever, cold, and all those. So, and then some of them will also go and form what we call the gametocytes, forming male and female gametocytes, okay? So these male and female gametocytes will then be in the blood, will then like be circulated in the blood. Then when another mosquito will come to feed on the person, they will like ingest this infected blood, and the cycle will continue inside the mosquito. Inside the mosquito stomach, the gametes, that is the gametocyte, will form a zygote, then the zygote will form what's called okinate, okinate will form what's called oocyte, and this oocyte will still give form the sporozoites. The sporozoites when it infects another person. So that's how the cycle continues. I hope you understand now. Dabi, can you hear me?
Yes, I understand. Better.
All right. So, anyone have another question? Go through it again.
So I think I have one more question for you, please.
Okay.
I just want you to I don't know, but do you have, aside this bacteriophage you showed us, I think you just lectured us on, I wanted asking, is there any other question your gut is telling you? Because I don't know, I have a feeling that maybe malaria life cycle. And then I think that, and okay, for me, just that. I don't know if anyone here has an idea, or maybe his or her gut is, giving you an idea of this question is going to go out. Now I'm asking everybody, please, if there is, can we just have an interactive class?
So, does anyone have anything that we can just
Well, all
um
I can say is I think um read your past questions. There are many. So in reading the past questions, you cover a lot of the material. So there is no way questions will not come out from even the past because they can't elude some parts. So
They can't elude some parts, such as, do you have an idea?
there is no idea here because my idea is failing me. But that's what I'm doing. If I combine her lecture, then I'm reading the past questions. I know there is no way question 1 to 4 or 5 will come out and I will say I didn't see what I wrote. It's not possible.
And that's another angle too. And if you notice any, like most of the things that maybe you read or you saw that they have not asked in the past question, just notice it very well. Or maybe when you come to all these viruses and you notice they've asked chickenpox very well or the rhinoviruses very well, that means you should read another one, master another one very well, another type of virus. Do you get? Because at this point, they tend not to repeat questions very well. That's what I've noticed. So it started from our own state. They have not repeated questions like that. So I just believe that no matter what, you're still going to pass. No matter what, you're still going to
Mama,
pass
please
as
give us
long as
a
you read.
clue on the
Yes.
Miro. Please give us a clue on the diagrams of all these viruses. They are at least what are the features that how will I put it? The diagram I think is up to 6 or 7.
Yeah.
So tell us maybe the common thing you can find in viruses so that even when we are missing points, we'll know, okay, this thing must be here, this thing must be here.
Okay, I think I mentioned it that day
Yes.
Yes.
of
Yes, you
virus.
did,
Or
but
plasmids.
it has vamoosed.
I beg, maybe follow you enter Hoye tomorrow.
I get this.
Let me check. See it here.
Call plasmid. Yeah, envelope spikes.
So this is just like a general structure of a virus, and the general structure they must contain either a DNA or RNA, that's nucleic acid. So you just have to note those ones that have DNA and then those ones that have RNA. Like in most of the virus structures, most of them has RNA, except the hepatitis B that has DNA polymerase. All right. Then for the capsid, capsid is a protein shell surrounding the nucleic acid. All the viral structure that we've treated in this material has a capsid. So you just know that if you are drawing any of the structure, just note the structure of a capsid. So I just draw the capsid for them. Then envelope, though is optional, but most of the ones we treated here, they have a cell envelope, okay? Then another one is what we call the spikes. The cell envelope, let me show you a picture of this envelope. So the viral envelope is just something that covers the cell or gives the viral structure a kind of a shape, or it protects it. Okay, that's a viral envelope. Then what we also have another one, spikes. Spikes are the glycoprotein that help in host attachment. You see all these things that is lifting up like how will I call it? Like a pin or something. See all these ones? Looking like it, like they are called spikes. They are glycoproteins. You can see in Ebola virus is called glycoprotein. Then HIV virus, they are also called glycoprotein. Then let me check if there's anyone they actually call it spikes. Okay, here they call it surface envelope protein. That is in dengue virus. Then here you call it envelope protein. So they have different names for it, but just know that the general name is the spikes or glycoprotein. Just use glycoprotein in case we are asked. So these are the four major the key structure in a virus. And then if you can remember other these things like hepatitis S antigen, hepatitis C antigen is an added advantage if you are told to draw the hepatitis B virus. All right.
They asked hepatitis B virus last semester, so I doubt they will this semester.
So yeah, it's just that even wrote it here. They asked us about hepatitis B virus. And funny enough, I cannot even remember it. I cannot remember it during the exam. I just draw, like I just remember I just picture, okay, most of the diagram are round shape with the spikes with the so I could not really I know that hepatitis has all these antigen, but I cannot remember where they are. So I just drew something and I know I drew something and they must give me mark for that thing I drew. So that's just it. So if you just have an idea of something, just write it. Even though you don't know that color that the main answer. If you just have an idea, please put it down for them. It's very important. All these small marks is better than empty sheet. The lecturer will not even know where will I even give this person mark or how will I even support this person. Okay. So but when you write something, oh, okay, she has an idea. Just take 5, take 10, take 20. Before you know it, you've passed the cutoff mark. All right. So is there any other thing?
Are we going to get this recording now?
Yes, it's available
All
now.
Thank you.
Thank you very much.
We really, really appreciate you. God bless you abundantly for your time, effort,
Honestly,
resources.
wishing you all the best. All the best,
Thank
all
you
Thank
the
so
you.
best.
Thank
much.
you. We are really
Pray
grateful.
for
And
us.
please,
Don't eat tomorrow.
don't leave
Thank
the
you very
Don't
exam.
eat
much.
tomorrow fast until we come out.
Yes, so it's very important. Please, don't leave the
You guys
hall. Don't follow others and leave the hall. You've really sacrificed a lot for you to just leave the hall empty. Just write
We'll come
something.
back to give you good testimonies
I
tomorrow.
beg, I can't wait.
Exactly.
Even I'm praying, God, I beg. Our effort should not be in vain in Jesus' name. Amen.
Amen.
Hey.
Good night. Thank you.
All right, good
Thank
night.
you.
Okay, no
God bless
problem.
you, ma.
Bless you too.
God bless you.